What is Life Recordings presents to you a visual summary of our most recent podcast on hybrids, as well as a further investigation of the CRISPR-Cas9 system. Our podcast investigates the world of hybrids both in plants and animal species.
What are you drawing?
A liger.
What's a liger?
It's pretty much my favorite animal. It's like a lion and a tiger mixed. Bred for its skills and magic.
A hybrid generally refers to any offspring resulting from the breeding of two genetically distinct individuals, which usually will result in a high degree of heterozygosity. In the wild, factors such as global warming can contribute to the creation of hybrid species. One of the examples being a griller or grizzly bear, which is a mix between a polar bear and a grizzly bear. Some of the issues resulting from hybrid species can be the ability to reproduce. In certain hybrids, there can be an imbalance of chromosomes coming from the two parents. A good example of this is a mule. A mule has 63 chromosomes, whereas a parent horse will have 64, and the other parent donkey will have 62 chromosomes. The mule has a very difficult time creating even unpaired sets of chromosomes called haploids, which results in unidentical pairs of chromosomes and renders meiosis nearly impossible. Infertility and other genetic defects can be a product in both natural and unnaturally occurring hybrids. However, there are many successful cases of biotic hybrids. In our podcast, we discuss some of these successes in maize crops. In this discussion, we address an important part of hybrid production, heterosis. Heterosis, or a hybrid vigor, is the increased performance of a hybrid relative to the parents and is a result of the variation that is present within a species. This is our segue into the main topic of this video, CRISPR-Cas9. So what is CRISPR-Cas9? CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. The repeats are short palindromic pieces of DNA, meaning they are 20 to 40 nucleotide long segments that read the same forward and backwards, just like the words kayak or race car. So we have a bunch of these repeats lined up, but instead of all being connected, they are separated by a spacer DNA, hence the regularly interspaced part of the name. These spacer DNA are different from the palindromic repeats in that they are all unique from one another. The curious thing that scientists discovered about spacer DNA is that each one matches up perfectly with viral and bacteriophage DNA. Bacteriophage are viruses that infect and replicate within bacteria. Scientists also found genes associated with CRISPR called CRISPR-associated genes, or Cas genes. The Cas genes create Cas proteins, which are proteins that unwind DNA, or nucleases, which are proteins that cut DNA. What they figured out is that this system is actually a way for bacteria to fight off bacteriophage. How this works is that usually, when a bacteriophage injects its DNA into a cell, it will create a bunch more bacteriophages and eventually kill the cell. But instead, with this CRISPR system, the cell is able to transcribe and translate proteins to create a Cas complex, while also transcribing DNA from a spacer protein to make CRISPR RNA that will fit right into the Cas protein. This fights the viral DNA by breaking the DNA apart, effectively stopping and ending the infection. This system also works when there isn't already an existing spacer DNA to match up with the DNA being injected. The CRISPR system is able to create a different class of protein from the one used earlier that takes in the DNA from the bacteriophage and breaks it apart. It then takes the DNA and copies it into the system as a new spacer DNA. So essentially what this CRISPR system is doing is keeping a history of old infections shown in the spacer DNA segments that help stop the cell from being infected again. Now that we understand how the CRISPR system works, let's take a closer look at the CRISPR-Cas9. The CRISPR-Cas9 was found when scientists were attempting to utilize the CRISPR system that they had observed in E. coli to either inactivate genes or to embed new genes. So scientists found this Cas protein in Streptococcus pyogenes, which is a species of bacteria, and called the protein Cas9. The Cas9 protein has a nuclease to cut DNA in two places, as well as two long strips of RNA. They have CRISPR RNA that fits into Cas9, as well as tracer RNA, which is connected to a spacer segment of DNA that matches up with the corresponding viral DNA, and which holds the CRISPR RNA in place. Together, the nuclease, the CRISPR RNA, and the tracer RNA form a complex that can break DNA. What these scientists did with this protein is that they put their own sequence of DNA into the spacer, or CRISPR segment, and then connected it to the tracer RNA. What this created was a tracer RNA CRISPR RNA chimera, which we can also just call guide RNA. So pretty much they created a new type of RNA and a two-part system made up of the Cas9 protein and the guide RNA. The guide RNA holds the information telling us where we want to make a cut in DNA, and the Cas9 protein is the part that actually does the cutting. So now what scientists can do is that if they have DNA they want to cut, they can create a guide RNA that has a corresponding bit of RNA. What happens is that the DNA will feed into it, and once in place it will be cut, thus inactivating the gene. The cell will try to fix the strand by creating mutations through insertions and deletions, but they can still effectively inactivate the DNA because they know exactly where to cut it. Let's say we want to make this more complex. Instead of just breaking a gene, we now want to insert a new gene. We will now need 3 parts: a Cas9 protein, a guide RNA, and a host RNA that we want to put in. As we break the DNA, the host DNA is going to be added, then the DNA is going to fix it, thus adding DNA to the cell. What's super cool about the CRISPR-Cas9 system is that it is able to do this in living cells and can make cuts in the DNA in multiple different places. So essentially, the CRISPR system is an immune system that we identified in bacteria and were then able to manipulate for our own human use. So the CRISPR is a very complicated gene editing system in our cells, but what could come of it? Why study CRISPR and devote research time and money towards a little immune response in cells? Well, everybody is so excited by CRISPR because it has seemingly limitless potential. We could do incredible things with it. For example, with the help of CRISPR, we could protect ourselves from viruses. That means things like HIV, herpes, and all other retroviruses. Although viruses do not hold a candle to cancer if we are talking about negative things that could be cured by CRISPR. Indeed, CRISPR may be the answer to making cancer a thing of the past. If we edit our immune cells to better detect cancer cells, then they would stop cancer very quickly before it became a problem. CRISPR could even be the answer to babies being born without genetic diseases. It would not even be that hard to fix genetic defects that are the result of only one miscoded letter in our DNA. With just a few shots, these awful diseases could be avoided. So say goodbye to muscular dystrophy. Aging could even be halted by CRISPR. So yes, CRISPR might even be the answer to eternal life. With a combination of genetic engineering and other therapies, we humans could end the aging, which is the greatest mortality risk factor of all. Although we are still a long way away from these progressions in science, CRISPR has the potential to change humans and the world in inconceivable ways.